Good morning, everybody. I'm Ken Takai, geomicrobiologist from Jamstack. It's my great honor to have a chance of talking at this ISA workshop. I greatly appreciate workshop organizer to give a chance to me. Today, I'd like to talk about microbial diversity and function in hydrothermal vent ecosystem of the Indian Ocean ridges. Okay, I like to start my presentation with brief introduction of research history and characteristics of deep sea hydrothermal system of the Indian Ocean. As most of you know, there are four major mid-ocean ridge systems in the Indian Ocean. From north to south, Carlsberg Ridge, Central Indian Ridge, Southeastern Indian Ridge, and the Southwestern Indian Ridge are present. These mid-ocean ridge systems are located in different, distant, and distinctive oceanographic and geographic regions, and geologically and tectonically so diverse. For example, Carlsberg Ridge and the Southwestern Indian Ridge are known to be typical ultra-slow spreading systems. On the other hand, Central Indian Ridge and the Southeastern Indian Ridge are known to be intermediate spreading systems. However, spreading rate and tectonic properties are so variable among the segments in these ridge systems. Then, the first deep-sea hydrothermal system of the Takai field was discovered in the Indian Ocean just 20 years ago. So Indian Ocean is the least explored ocean for research and exploration of deep-sea hydrothermal systems. In 1993, Japanese research vessel Hakoma conducted hydrocast plume survey over the Central Indian Ridge. During this expedition, Japanese team successfully detected geochemical signature of hydrothermal plume at the Central Indian Ridge near the Rodriguez Triple Junction. Based on the geochemical signature, in 1998, 3 dives of human-occupied vehicle Shinkai 6500 was conducted by Japanese research vessel Yokosuka. However, this expedition did not find any evidence of the seafloor hydrothermal activity there. However, in 2000, Japanese research vessel Kairei expedition was conducted again. Finally, remotely operative vehicle Kaiko successfully discovered the seafloor hydrothermal activity, the Kairei field. After this discovery, Japanese team published 2 important papers about geochemical characteristic of high-temperature hydrothermal fluid and the general composition of the chemosynthetic macrofaunal community of the Kairei field. About 6 months later, American research vessel Knoll and ROV Jason expedition was conducted at the Central Indian Ridge in the similar places to the previous Japanese expedition. During this expedition, American team discovered second deep-sea hydrothermal system in the Indian Ocean Edmont field and revisited the Kairei field. Then they discovered very unique Indian Ocean deep-sea hydrothermal vent endemic gastropod, so-called scaly-foot snail in the Kairei field. This gastropod has unique morphological feature, scale on the foot body, and iron sulfide biomineralization armor on the wall surface of the body. In addition, this expedition provided the first brief description of microbial diversity and function in the Indian deep-sea hydrothermal systems. Just for your information, very recently, I knew of a surprising fact that, according to the paper, scaly-foot gastropod was discovered by American expedition. But in 2000 Japanese expedition, the Japanese team already found living scaly foot gastropods and obtained several individuals. But first description and discovery was conducted by American team and their colleagues. So I guess there is something happened. Anyway, now 13 major active deep-sea hydrothermal systems are discovered and confirmed in the Indian Ocean. The most important point is very recently, Chinese research group have successfully discovered many deep-sea hydrothermal systems in Carlsberg Reach and southwestern Indian Reach. An increasing number of deep-sea hydrothermal systems has been discovered very recently. Among these newly discovered hydrothermal systems, not so many microbiological characterization of the entire microbial diversity and function has been conducted. Actually, a detailed characterization was conducted in this field, and most of them came from my research group. This is the reason why I do presentation today. Anyway, this is a brief conclusion of my presentation today. The question is, are microbial diversity and function variable in different hydrothermal systems of the Indian Ocean? The answer is, of course, yes. But it's not just for the Indian Ocean system, but for any of the global hydrothermal system. Because I already concluded that microbial diversity and function are principally controlled by microbiologically available energy mass balance in the mixing zones of habitats between hydrothermal fluids and the ambient seawater, and not necessarily controlled by biogeographic evolutionary isolation, partition, and disconnectivity. So next, I briefly talk about this conclusion. Now, many people know that more than 500 deep-sea hydrothermal systems has been discovered all over the world. And these deep-sea hydrothermal systems are geographically, geologically, and geochemically so diverse. Many people also know that chemosynthetic, microbial and macrofaunal community are strongly dependent on the chemical energy input from the hydrothermal fluid. So now you can intuitively understand these arguments. The geological diversity of hydrothermal system makes chemical diversity of hydrothermal fluid. And the chemical diversity of hydrothermal fluid makes metabolic diversity of chemolithotrophic microbial community. Finally, metabolic diversity makes the compositional diversity of chemolithotrophic microbial communities. These arguments seems to be very reasonable, but this was for the first time formulated by some dynamic estimation of microbiologically available energy mass balance in the hydrothermal mixing zones habitats by McCollom-Shock in 1997. I always call this as McCollom-Shock's prediction. Here, I briefly explain about McCollom-Shock's prediction. They first calculated available gives free energy change of each of the chemolithotrophic metabolism in the hydrothermal mixing zone of habitat, as you can see in this figure. Then they predicted the energy mass balance would reflect the realistic composition of the chemolithotrophic microbial community. Their calculation was based on a single type of deep-sea hydrothermal system, such as mid-ocean ridge basalt-hosted black smoker type. But their calculation seems to be very applicable to any type of deep-sea hydrothermal system all over the world like this. However, no one succeeded in obtaining the direct evidences. Actually, we tried to justify McCollom-Shock's prediction by investigation of many deep-sea hydrothermal systems all over the world. This table shows physical and chemical properties of typical high-temperature hydrothermal fluid in representative deep-sea hydrothermal system. Finally, we compiled these data for 91 fields from all over the world. Then we conducted McCollom-Shock's prediction for 91 fields. Then this is a results, and I'm sorry, this is very messy figure to see. But important point is very, very simple. Different deep sea hydrothermal system would host different composition of energy metabolism and microbial population. Another important point is the deep sea hydrothermal system with similar geological setting. For example, basalt-hosted type, ultramafic rock-associated type, sediment-associated type, and submarine volcano type have similar composition of energy metabolism and microbial population. But this is still just prediction. Next week, we compared theoretically predicted microbial community composition and experimentally determined microbial community composition, showing pie graph and bar graph, respectively, in representative deep sea hydrothermal system, mainly using a quantitative cultivation technique and a genetic analysis. And I'm sorry again, this is a very messy figure to see. But in this figure, the same color show the similar energy metabolism in a similar microbial population. Then you can see both theoretically predicted and experimentally determined microbial community composition have similar color pattern, similar composition. So these results strongly suggested that some dynamic estimation of microbiologically available energy mass balance can predict realistic composition of the chemolithotrophic microbial community in the deep sea hydrothermal environment. So McCollom-Shock's prediction was justified by our investigation. In other words, we successfully found one of the basic principles to control the chemolithotrophic microbial community development in any type of deep sea hydrothermal system all over the world. So according to McCollom-Shock's prediction, newly discovered deep sea hydrothermal system in the Indian Ocean without detailed microbiological characterization can be easily predicted like these. In the most of the ultramafic rock-associated hydrothermal system, microbial community would be predominantly supported by a major hydrogen-trophic microbial population. In other basalt-hosted systems, the microbial community would be predominantly supported by major cytotrophic and minor hydrogen-trophic microbial population. Actually, this is one of the conclusions in my presentation today. Finally, I'd like to show you two new discoveries in the Indian Ocean. One is about endemicity and microevolution of the cosmopolitan chemolithotrophic species in the global deep sea hydrothermal system based on the study of my Japanese colleague, Mino-san. They collected several cosmopolitan chemolithotrophic microbial species and strains from different deep sea hydrothermal systems all over the world. Then they compared the genetic distance and the evolutionary rate of these chemolithotrophic microbial species. Then they found that heterogeneity in the evolution actually occurred in the several cosmopolitan chemolithotrophic microbial population in different deep sea hydrothermal systems all over the world. So these results strongly suggested that there should be working biogeochemical control of microbial diversity and function, although it's still just a minor impact. Another discovery is serpentinization-driven low-temperature hydrothermal system, Old City Field, Indian Ocean. As most of you know, serpentinization-driven low-temperature hydrothermal system was first discovered in the Atlantic Ridge, Lost City Field. In 2001. The later similar system was discovered in the Pacific, particularly Mariana Trench, Fort Egg region, Shinkaishi Field. Actually, the geological characteristics and the geochemical feature of hydrothermal fluid, Old City Field, are still unpublished. But microbiological community in the several carbonate and blue site chimney of Old City Field were reported this year. According to the results, several blue site chimney hosted very, very unique microbial community, strongly dependent on the carbon monoxide and formate, not dependent on the molecular hydrogen and carbon dioxide. Due to the strong alkalinity of hydrothermal food, strongly inhibiting availability carbon dioxide and bicarbonate, and chemolithotrophic hydrotrophic function. Actually, this new discovery provided new information about hydrothermal type and microbial community in the Indian Ocean deep sea hydrothermal system, previously unexpected by McCollom-Shock prediction. And of course, there are so many hydrothermal systems are still unexplored and discovered in the Indian Ocean. So further exploration will provide a new insight into understanding the microbial diversity and function in the Indian Ocean deep sea hydrothermal system in the future. Okay, this is the end of my talk. Thank you very much for your attention.